I'm surprised. We are more than 10 at the end of this meeting, which is a good sign. And I thank you all for staying so long. Clearly, from the standpoint of ARIS and hopefully from the standpoint of the other organizers, I think that the meeting has fulfilled our expectations. Namely, it sparked very stimulating and constructive discussions between both naturally public and private players. And hopefully it helped establish potential partnerships through the many B2B meetings that have been organized. Basically, we have filled most of the slots that were planned. So the meeting was also a great opportunity to highlight the excellence of the French researchers and innovators in the field of cell and gene therapies, thanks to the excellent overviews and pitches on upstream and downstream topics, thanks to the very active roundtable discussions as well as presentations of several assets of the French ecosystem that make it particularly conducive to the implementation of cell and gene therapies. So I won't have time to summarize here all the topics that have been discussed, but we'll mention a few bullet points. So regarding AAV and lentiviral therapies, as well as gene replacement or editing approaches, several excellent centers in France gathering translational expertise covering all the aspects of product development have been presented. Several shared challenges regarding costs, specificity, efficacy, toxicity, immunogenicity, lack of relevant preclinical models, and with a recurrent issue regarding cell and tissue targeting, as well as toxicity associated in particular with off-target effect of some gene editing technologies, have been extensively discussed, and I think in a very productive and constructive fashion. There have been also a number of promising perspectives that have been highlighted, such as improved AAV vectors through chemical modification, computational design, and through harnessing of AI-generated libraries. Also, implementation of DSB-free gene editing approaches with clear opportunities to increase potency and specificity, decrease doses and toxicities, as well as manufacturing costs. Regarding mRNA-based clinical application, these were nicely reviewed by Chantal Pichon, and the ensuing discussion on siRNA and oligonucleotide strategies, in particularly during the roundtable of this session, highlighted not only several recent successful achievements with mRNA vaccines and siRNA, but also here again recurrent challenges dealing in particular with specificity and targeting, as well as opportunities to better leverage AI/ML-based approaches to improve design of the vectors and these approaches. Regarding immune cell-based therapies, mainly CAR T cells, so these, as you know, have yielded many promising clinical results in both oncology and more recently in autoimmune diseases. But there are here again many challenges and unmet needs. In particular, how can we apply this approach to tackle solid tumors? How could we use allogeneic CAR T cells to reduce costs, improve standardization, and scaling up? Many other issues were largely discussed during the roundtable regarding efficacy, which depends on the definition of the right target antigen, the control of tissue homing, survival, effector function, ability to thwart tumor escape mechanism, as well as toxicity, natural toxicity issues. So given the diversity of unsolved issues, all the participants, and I think also the audience, agreed on the importance to involve all relevant experts from both the academic and private side, but also patients and regulatory players to move forward efficiently. Finally, regarding non-immune cell-based therapies, so while promising clinical data have been obtained here again, discussion focused on the actual mode of action, in particular of MSCs, seemingly primarily through supportive or endocrine activity rather than regeneration. Also, so there was discussed the need for robust qualifying and potency assays and the respective caveats and advantages of cell versus exosome-based therapies. An important point that was raised was the importance to follow up the patients and share data, clinical and real world, ideally, for the benefit of the whole community to accelerate cell and gene therapy development and allow accurate assessment of the cost-benefit ratio of these approaches. So just to finish, I would like to, and most importantly, acknowledge a number of people who have been key in the organization of this meeting. So naturally, Stéphanie, Véronique, and Jean-Pierre from Aris, Violaine Gauthier and France Biotech, Nathalie Manot and Delem. Nathalie did really a fantastic job. Thank you, Nathalie. Rosalie Maurice, Julie Lelouch, and BPI France. Franck Le Timonier, thanks a lot, Franck. You were instrumental in the success of this meeting, and as well as other ITMO directors and the whole INSERM teams. Jean-Marc Gauthier, Hervé Coste, and Gloria Baton from INSERM Transfer, who were of strong support throughout the organization of the meeting. Several students from Sud Biotech, Amanda, Matteo, Océane, and Yasmina. Fabrice Papillon, thanks Fabrice. Thank you. It was a very dynamic discussion. People were impressed and thought that you were a scientist. You are a scientist, almost.
No, but I could turn into.
Yeah. I would like also to thank on behalf of the organizers all the academic, biotech, and pharma speakers. You did fantastic presentation, fantastic discussion, and naturally all the RT roundtable participants. We would like also to thank the excellent, the AIS and SGPI, and in particular Lise Alter and Bruno Baudel, who did a really nice presentation of the French ecosystem, as well as the Ministry of Economy and Finances, who kindly hosted the meeting and in a fantastic environment. So thank you very much. Have a good trip back and hopefully establish many, many fruitful collaborations. Thank you.